Once the sound stimulus has been transduced by hair cells in the cochlea of the inner ear, the information then must be relayed up to the cerebral cortex for conscious awareness. Now, the pathway for audition has a few more stops along the way than the somatosensory pathways we've discussed before. So I want to orient you to what we are looking at. So these are obviously the two ears, and we have on the right side of the head here, we have all the structures of the outer, middle, and inner ear. And that's shown in blue, and we have similar for the left ear over here in red. And so remember that it is the auditory nerve, also sometimes known as the cochlear nerve, cranial nerve number 8, that is going to be monitoring, that consists of neurons that monitor the activities of the hair cells in the cochlea. And so they are going to relay the information directly into the brainstem to a nucleus. They're paired, one on the right and one on the left, called the cochlear nucleus. So from the cochlea, we go to the cochlear nucleus. And the cochlear nucleus consists of neurons. A nucleus, remember, neuron cell bodies and dendrites, information processing center. It's going to process information coming only from that one ear. From the cochlear nucleus, and again, right ear shown in blue, left ear shown in red, the information will then be relayed to a nearby nucleus in the brainstem called the superior olivary nucleus. And there's one on the right and one on the left. Now, what happens is the superior olivary nucleus gets information from both ears. So it gets information from the ear on the same side, and it's also getting information from the cochlear nucleus on the other side. So the superior olivary nucleus in the brainstem is the first place where neurons in the auditory pathway are getting information from both ears, and they're starting to process it together. From the superior olivary nucleus, the information then travels up to the midbrain, to the inferior colliculus, which are these bumps on the back of the midbrain. Remember, there's two pairs of bumps on the back of the midbrain. The lower bumps are the inferior colliculi. There's one on the right, one on the left. And these structures, obviously, again, information processing center, they get information from both ears because they're getting the information from the superior olivary nucleus. They are very important in auditory reflexes, like the startle reflex. If you're sitting in class and someone slams the door behind you and you involuntarily jump, that is a startle reflex to auditory stimuli that is handled by the inferior colliculus. When a noise is really loud and you turn your head away from it, that's an auditory reflex handled by the inferior colliculus. Again, these structures are getting information from both ears. From the inferior colliculus, information is then relayed to the thalamus, and the specific nucleus of the thalamus that receives the information is called the medial geniculate nucleus. You do need to know that. And it is dedicated to processing information only from the auditory system. From there, neurons in the medial geniculate nucleus of the thalamus will relay the information finally to the primary auditory cortex in the temporal lobe. And this is, and there's one of course on each side, when the information arrives and is processed by neurons there, the sound information will be processed, you'll become consciously aware of what you're hearing, and you will be able to recognize what you're hearing. So this is a kind of a complex pathway to remember the names of all the structures and where they are in the brain. So I want to give you a little assistance. So we go from the cochlea to the cochlear nucleus. That's the first stop, and that is in the brainstem, and specifically the medulla oblongata. I'm sorry, the pons. That is an important thing to know. Now, From there are two more stops in the brainstem, and you have to remember that in each place, each of these nuclei, there are synapses. So neurons from the cochlear nucleus are going to actually synapse with neurons in the next stop, the superior olivary nucleus. And then those neurons are going to synapse with neurons in the midbrain, the inferior colliculus neurons. So we have cochlear nucleus, superior olivary nucleus, inferior colliculus. If you remember the mnemonic device, Sonic MG, you will always remember that the stops in the brainstem are the superior olivary nucleus, SON, then the inferior colliculus, the IC, and then the reason that there is a space, and this is a separate word, is that we are now leaving the brainstem and going to the medial geniculate nucleus of the thalamus. And then always from the thalamus we go to the primary auditory cortex. So you just have to remember the cochlear nucleus is not part of this mnemonic, but obviously from the cochlea we're going to go to the cochlear nucleus, then superior olivary nucleus and inferior colliculus, both of which are in the brainstem, then medial geniculate nucleus of the thalamus, and then to the primary auditory cortex. So this is the primary neural pathway for audition. Very important that you know this, study this, add this to the somatosensory pathways that you've already learned. So once we get up to the primary auditory cortex, just like the primary somatosensory cortex was somatotopically organized, so neurons in specific places were dedicated to processing information coming from specific body parts, we have a tonotopic map, a frequency map in the primary auditory cortex, so certain neurons are dedicated to processing certain frequencies of sound. And this is in kilohertz, so this is in thousands of hertz, so toward the back of the primary auditory cortex, the highest frequencies that we can perceive, 20,000 hertz, 18,000 hertz, there are neurons that are dedicated to processing that, whereas as you move toward the front, the lower frequencies have neurons dedicated to processing them. So there is actually a tonotopic map in the primary auditory cortex. Now, one of the most important things that we do with auditory information is we are able to localize or figure out where in space the source of a sound is. And this is particularly important when we're in a loud, noisy environment where there are a lot of different sounds going on and you are trying to localize the voice of your friend that you are trying to meet. You can't see them, maybe you're hearing them. Also, it helps us figure out where things are in space so that we can either go toward them or avoid them if they're, for example, sounds of a predator. So the way that this works is I want to go back to the map of the auditory pathway here, because remember that from after the cochlear nucleus, every nucleus along the pathway is getting information and processing information from both ears at the same time. And that's very useful in sound localization, because the cues that the auditory system of the brain uses to figure out where a sound source is located has to do with information that comes from both ears and comparing them. So let's consider that we have a person and the source of the sound, like their eyes are closed, they don't know where the sound source is, and we ask them to localize it in space. They will pretty accurately localize it as being to the left and in the front of their head. How do they do this? So recall that the source of a sound is going to create alternating waves of high and low pressure. There are two different cues that this leads to. One is that the head creates a barrier to this sound. So sound waves can't travel, through the head. They have to go around. So this means that the loudness of the sound, or the intensity, is going to differ between the two ears. The ear that is closest to the sound source is going to hear the sound more loudly than the other ear, because as sound travels with distance, by the time the sound gets past the head and over to the right side of the ear, it's not going to be as loud because it's just traveled further, and it's going to fade with distance. So there are sound loudness or sound intensity differences between the left and right ear, and what happens is the brain, particularly the superior olivary nucleus, it's the first place this happens, but other parts of the auditory pathway as well, they compare that difference between the loudness of the sound at the left and right ear and can predict based on that exactly where the sound is closest to, right, left side of the head. We have the same issue in terms of time of arrival. So again, it's going to take a longer time for the sound to reach the ear on the other side of the head than on the side of the head where the sound source is. And that difference in latency, or the time of arrival, the difference between when the sound gets to the two ears, is the second cue that the auditory system is going to be able to use to localize the sound source. So there's a lot more to sound localization than that, but I want you to understand that this is the key and primary information, and that if you don't have hearing in one ear or another, it can be more difficult to localize the source of a sound in space. So whenever we talk about hearing, we always want to talk about the lack of ability to hear, which is referred to as deafness. There's a number of different causes. The most common cause is what's known as sensory neural deafness, and it's usually a problem in the cochlea with the basilar membrane. There's usually some kind of genetic factor or some kind of environmental factor that has prevented the basilar membrane from developing properly or the hair cells from developing properly. And so literally transduction can't take place. So everything up to the cochlea is happening fine, and everything in the brain would be working fine, but the cochlea itself is not able to transduce the sound stimulus due to a problem with or lack of hair cells. And this is where cochlear implants can be used to restore some degree of hearing. And your textbook talks about cochlear implants and exactly how that works, basically creating kind of an artificial basilar membrane that can distinguish between frequencies and then send the signals up to the brain, which is working just fine. A less common type of deafness is what's known as conduction deafness, and this is if there is problems with anything leading up to the inner ear. So maybe you have earwax clogging the auditory canal. Maybe you have an ear infection where instead of just air in the middle ear, you actually have fluid buildup due to inflammation, because this is connected to the throat and all the sinuses. So if you have an infection, you could get fluid in there that prevents the tympanic membrane or the auditory ossicles from moving properly. You can also get arthritis in the joints of the auditory ossicles. You lose the elasticity with age of the tympanic membrane. These can lead to conduction deafness. So there's a problem with getting the sound to the inner ear. And most of these are not super severe, don't lead to total deafness. And in the case of the ducts being clogged or inflammation in the middle ear, obviously that's a temporary situation. Another kind of deafness is known as central deafness because it's in the central nervous system, meaning the brain. There's something wrong with the structures in the auditory pathway of the brain, cochlear nucleus, superior olivary nucleus, inferior colliculus, medial geniculate nucleus, or the primary auditory cortex. There could be problems in any or all of those areas that even though the sound is properly conducted and transduced, it is not able to be transmitted to the proper areas of the cortex for conscious awareness. This is a more rare kind of deafness than problems with the cochlea. Now, what does happen over time and with exposure to loud environments, anything over 85 decibels for long periods of time, is that the hair cells in the cochlea start to die and decline. So if we were to look at a healthy normal cochlea, and the hairs and the hair cells on the basilar membrane of the cochlea under a microscope, this is what it would look like. You can see all these nice, lovely hairs and a lovely structure of the cochlea. And under an electron microscope, we could actually see individual hair cells themselves, that just the hairs protruding out of the organ of Corti. You can see how nice and healthy they are. In a person who has experienced severe noise damage, this is what the cochlea would look like. You can see there are large regions of the basilar membrane where there are literally no hair cells. And if you look at the electron microscope, you can see that there are areas where there are no functional hair cells, or where the hairs on the hair cells are just really, really unhealthy and crushed and sickly. This is not reversible. Hair cells, while they are not neurons, they do not regenerate. So you want to try to preserve your hearing as much as you can. Over the course of the normal human lifespan, we are just exposed to all sorts of normal human noises, and we do lose some hair cells as a normal course of aging. This is known as presbycusis. There are also drugs that certain kinds of antibiotics, they're not commonly used, but they can have a side effect of damaging hair cells. But it's typically due to noise exposure over time. And we lose not all of our hearing, but we lose the hair cells that really transduce kind of high-pitched sounds as well as low-pitched sounds. We can get more severe hearing loss at much younger ages if we are exposed to continuous chronic levels of noise that are over 80 decibels. And this includes noise exposure that can come from your job, occupational noise, or from leisure. So use of power tools, lawnmowers, guns if you're not wearing ear protection. If you work at an airport, obviously on the tarmac, you need to wear hearing protection. Most sporting events, if you go to a Ravens game, the sound of the crowd and the announcer and the music that's played through the loudspeakers well exceeds the safe level of 85 decibels, and you're there for 3 hours. So you might have a ringing in the ears that's caused from, damage to some hair cells. Also, playing in a band or going to hear a live band and standing right next to the speaker. So these are just examples of some of the types of environmental noise that can cause, over time, death of hair cells. And one of the things that can happen is, and they think it's from death of hair cells, is this persistent ringing in the ears and loss of hearing for certain frequencies called tinnitus, which can be extremely, everybody has it to some degree. If you have ringing in your ears, sometimes to some degree it's a normal thing. However, if you have it all the time, chronically, it can be really distracting, and it can also impair your ability to hear certain frequencies. There's no cure for it right now. It's being studied, and it's thought to be a result of the death of hair cells. So you want to try to preserve using, being exposed to loud noises for long periods of time, without any kind of ear protection. And I will just mention that iPhones and other kinds of listening devices, typically if you go to the max volume, are well above 85 decibels.